Assalamualaikum and greeting. My name is Intan Nur Izzati binti Roslan, a final year student of Bachelor of Science Honour in Biology. Today, I am going to present my final year project with the title Diversity and Abundance of Mosquitoes, which is Nematocera at two different elevations of Cadamba Trail, UiTM Puncak Alam, under the supervised Dr. Siti Khairah binti Muhammad Hatta. Background of the study. According to pattern 2017, insects are most successful group of animals in the world. Based on Garvey 2018, insects can survive in a wide range of conditions, from hot springs to freezing temperatures. Any insect that has two wings or so-called true fly are commonly from order Diptera. Diptera can be classified into Nematocera and Brachysera. The suborder Nematocera is an insect with multi-segmented antenna and has a thin body with long legs such as cranefly, midget, genet, and mosquitoes. To date, there is no study on insect diversity has been conducted in UiTM Puncak Alam. Problem statement. The rising development and human population around Puncak Alam lead to deforestation that later can threaten the diversity of organisms including Nematocera. This is due to the biology of Nematocera that depends on the vegetation in their life. Deforestation often leads to habitat loss that later change the food chain in that habitat. According to WHO 2022, nearly all decreases and extinction of insect population are caused by human action. Therefore, a study must be carried out to serve as baseline data for future references to understand the impact of human activity towards the diversity and abundance of Nematocera, especially in the Puncak Alam area. Objective of this study. First one is to determine the diversity and abundance of Diptera species in Cadamba Trail UiTM Puncak Alam. Secondly is to compare the diversity of order Diptera from two different elevations in Cadamba Trail of UiTM Puncak Alam. Methods. Setting Malai trap. Insect sampling was conducted in Cadamba Trail UiTM Puncak Alam by placing Malai trap at two different elevations which are low and high elevations. Second, sorting and pinning. After 14 days, all the samples were collected and sorted according to their respective orders and family. The samples collected were pinned using an entomological pin. Next, order identification. The samples were identified using reference book, website, and journal articles. All the data were analyzed using Shannon-Wiener Diversity Index, Evenness Index, and Margalef Index. Overall result, there are 1,101 total individuals were collected, consists of 8 families. From those 8 families, 51 morphospecies were identified. From the overall sample collected, 501 individuals were collected from the low elevation and 600 individuals were collected from the high elevation. Cecidomidae contribute to the highest family found, which represent 34%, followed by Ceratopogonidae, which is 28%, and Ciaridae with 16%. According to Dorchin 2008, Cecidomidae are one of the largest group of Diptera, consisting of 5,700 species described and known as a pest to agriculture, food crops, forestries, and ornamental plant. Next, family Ceratopogonidae is the large family with approximately 4,000 species and important in medical and veterinary entomology and colonize high altitude area. According to Vogel and Martin 2000, Ciaridae act as a pollinator. They are considered as ineffective pollen carriers due to their tiny body and weak flying ability. Pollination by Ciaridae has been seen in 8 angiosperm families. The least common family of Nematocera found was Psychodidae, which comprises 0.01%. Psychodidae is a homometabolous insect that live their whole life in aquatic, semi-aquatic, or terrestrial habitats, and adults consume plant items like nectar. The females of certain species and some males feed on blood. For the discussion regarding the abundance of Nematocera, the highest number of individuals were collected at Trap 2 with the total of 600 individuals, followed by Trap 1 with 501 individuals. The value in the bracket is the total number of morphospecies among families of Nematocera at study site. Nematocera found more abundance at high elevation compared to low elevation. High elevation is receiving more sunlight, which cause high temperature. Nematocera are more diverse since they are drawn to areas with more sunlight. According to Code Blue 2022, rising temperatures support the survival of the insect and extend territories. Abiotic factor control the ecology of insect colonies. This statement was claimed by Savopolo 2012 that insect abundance and distribution are regulated by biotic and abiotic factors. Meanwhile, at low elevation, Nematocera found were less abundant. This might be due to traps placed at low elevation located near the entrance of Cadamba Trail. The location near the entrance might be prone to disturbance such as deforestation. This is because the location is widely open and exposed to human activities. Now, we move on to the diversity, evenness, and richness of the Nematocera. Diversity and abundance of Nematocera were calculated by using Shannon-Wiener Diversity Index, Evenness Index, and Margalef Index. The Shannon-Wiener Diversity Index in Trap 2 has the highest family diversity with a value 2.987, followed by Trap 1 with a value 2.763. It demonstrates that Trap 2 is more diverse compared to Trap 1. According to past research done by Muhammad Hatta 2015, when H values is less than 3, it is suggested that the research area was disturbed and unstable. The evenness index at high elevation recorded a higher value than at low elevation with 0.484 and 0.453 respectively. This indicates that the distribution of the species at the high elevation was more evenly distributed, and there are dominant family in Cadamba Trail. The highest value of Margalef was in Trap 2 is 6.253 and Trap 1 is 5.469. Many species richness depends on the sampling effort. Further analysis using Mann-Whitney showed that there was a significant difference between the distribution of Nematocera between the low and high elevation. Value of H is correlated with the value of R and E. According to the 2018, when the value of E is approaching to 1, it indicates that the habitat have evenly distributed individuals. Conclusion: based on the analysis of the study, the most diverse Nematocera at high elevation may due to the abundance of sunlight. High elevation has the most species richness with a value 6.253 and evenly distributed with a value 0.484. Low elevation has less distributed due to the area near the entrance, sampling period, rainy day, and the number of Malay trap used. The result of this study did not support the hypothesis that Nematocera have similar diversity and abundance at low and high elevations. For the recommendations, to get better result for the inventory of Nematocera, the number of Malay traps should be increased. Next, increase the sampling period and study site. Lastly, integrate climate because climate influences the diversity and abundance of Nematocera. This is the list of references. Thank you.